All right. Hello everyone. Welcome back to the WebEVTalk series on the 24th August 2021. And I'm really excited today that we have Professor Marca Wauben, a very well-known prominent researcher in the extracellular vesicles field, to join us today and to share her work in breast milk. So Marca is a professor of intercellular communication at Utrecht University with a background in biology. Her research team aims to unravel the role of intercellular communication in biological systems via extracellular vesicles, with specific emphasis on the regulation of the immune system. They use multi-omics approaches and employ high-resolution flow cytometry-based methods for EV characterization. And they also aim to identify immune regulatory vesicles in body fluids, for example, in breast milk, blood, synovial fluid, and try to reveal their origin, their cargo, and pathophysiological roles. So besides the fundamental research, the lab works on EV-based biomarker discovery and therapeutic EV applications, and the development of nanotechnology to analyze EVs. So today, her lecture is titled Deciphering the Physiological Role of Extracellular Vesicles in Breast Milk. And before we start, I'd like to thank Horibai Scientific, our sponsor for this session. And if you're interested to support WebEVTalk, please go to the patreon.com and find WebEVTalk or direct message us.
Again, thanks, Carolina, for this very nice invitation. And today I really would like to share some of our published, but also of our very recent results on extracellular vesicles in breast milk. And that's all because we are really trying to figure out what is the physiological roles of these vesicles in breast milk. So before I start my lecture, I would like to disclose that part of the work has been done in collaboration with Danone Nutrition Research in a collaborative partnership grant, and with Friesland Campina in the Netherlands. So for this audience, I think it's no need to really explain what extracellular vesicles are, and but it is always good to recap. And I think from my point of view, it's extracellular vesicles are multi-component regulatory platforms that are released by cells in the environment. And there are, of course, some common protein signatures and lipid signatures, but also selective incorporation of cargo in these vesicles. And because of this selective incorporation of cargo and this strict regulation, it is clear that a different composition will, in the end, lead to a different function. And it is also obvious, of course, that there are many different EV subsets. For this talk, I think it is important also to address the point that several of these subsets appear to be quite stable and that they can cross classical epithelial and endothelial borders. If we talk about EV-mediated communication, it is clear that in all biological systems, in all kingdoms of life, EV-mediated communication takes place. And it is not only communication within an organism in which these EV take an important position, but it is also the communication between different organisms in which these EVs can have a very nice role. And in that respect, we are very interested in deciphering the physiological role of these EVs in breast milk, because that's the way how mother can give some information to her child. And we all know that what you eat in the first days makes really a difference for the rest of your life. And in that sense, milk is nature's first functional food. Milk contains many factors that are involved in the maintenance of immune homeostasis and building the immune system and prevention of inflammation and disintegration of the barrier. So what we investigate is whether milk EVs can be a modest way to show or to educate a newborn how to cope with a novel environment. And before going to the milk EVs, I would like to draw the attention to breast milk in general, because breast milk is a complex living fluid. In breast milk, there are many different cell types present, like epithelial cells, neutrophils, lymphocytes, but also microbiota, living microbiota is present in breast milk. And as such, all these cells can also release different components in milk. And what you can see in milk is that there are also specific colloidal milk structures, like the milk-fat globules, also different sizes, and of course the extracellular vesicles, but also the casein micelles, and also the aggregates of the whey proteins and specific milk oligosaccharides. So that makes it really complicated to really study the functional effect of only one structure in milk. So already in 2014, we started to work on a protocol how to recover extracellular vesicles from human breast milk. And in this publication in JAV, we showed that the recovery of the EVs from human breast milk is highly influenced by the way of sample collection and sample treatment, and also by the vesicle isolation procedures. Without going into full detail on it, because it is all published, I quickly run through the protocol that we use. We always start with fresh milk, which is kept at body temperature and is processed very rapidly in the first, within 30 minutes to 1 hour. And then differential centrifugation protocols are performed. And what is interesting is that we developed a protocol in which we loaded the 10,000 Joule supernatant on top of the density gradient to avoid all kinds of problems in the pellets due to caseins and other milk components. So we developed the protocol and then isolated the EVs from the different density fractions. And recently we further worked on this protocol, and that is published in the JAV paper in 2021, indicated here, in which we, after the density gradient, performed a size exclusion column step, in which we collected in the end the EVs in a concentration which is more or less similar than the original concentration in milk, meaning that we performed the in vitro assays on cell modulation assays. We performed that in the concentration in which EVs are normally present in milk. Besides that, we also developed a real procedural control to really see whether the effect that we measure is truly evmediated and is not due to the procedure or to the milk context matrix as such. So by using that protocol, we then next characterize these milk EVs, and what can be seen here already is that these milk EVs, if we look to cryo-EM, are still very heterogeneous in size. They are even not always round. And also in composition, because here is a milk EV depicted that is fully decorated with an XN5 to show that the PS is flip-flopped to the outer layer of the lipid membrane, while other vesicles do not stain with an XN5. So despite having that protocol to isolate all these nice EVs, you can still see that it is very heterogeneous. What I think is quite remarkable, and that we published in the recent JAV paper now, is that if you analyze the amount, the quantity of EVs in mature milk samples, So these are mature milk samples of an average of 7 months postpartum, so running roughly from 3 months on. Then the amount of EVs is quite similar, and it's very high, because when you look here to the particles per ml, it's roughly 1 × 10⁻11 particles that are present in milk. And in our hands, when we use the same strategy for the isolation of plasma EVs, milk EVs has roughly 10 times more EVs than in plasma. If you look to the size of these milk EVs, they also tend to be a bit bigger than the plasma EVs that we isolate. And also here you can see that they are quite heterogeneous. For the common markers to detect on Western blot, they are positive CD9, CD63, we can detect fertilin-1 and also the heat shock proteins. So after we had characterized these, after we isolated these EVs, then the next step was to perform a proteomics analysis of these EVs. And my group published in 2016 that by using this protocol, we could unveil a novel functional proteome that was clearly distinct from other milk components. In that study, we showed that all the proteins that have been described in human milk are collected here in blue, that the proteins that we discovered in the isolated milk EVs, here indicated in green, that more than 600 proteins were never described in the milk proteome before. And so now the next step that we took was to see whether we can connect somewhere the milk proteome with functionalities of these milk EVs. And the first step Martijn did was to see which proteins are present in all milk donors. Like here we have 7 milk donors, and in all these donors 367 proteins were present. And we use these 367 proteins to perform enrichment network and functional annotation analysis. So when you think about the function of milk EVs, the first encounter of milk EVs in the newborn is in the oral cavity. So the first question we pose is, do these milk EVs interact with epithelial cells, and can they modulate the oral epithelial cells? For that, we used a model system, the oral gingival CA922 cells, and we performed a gap closure assay. In this gap closure assay, when we added milk EVs, we could see that there was an accelerated gap closure when you compare it with the procedural control here. And this is also depicted here and quantified. The next step was then, how can we connect this enhanced gap closure to the proteome of the milk EVs? First, if you think about enhanced gap closure, that can be due to either enhanced migration of cells or enhanced proliferation of cells. What is depicted here is the 2 pathways, the hotspots in these 2 pathways, and in blue are indicated the milk EV proteome. So what is already clear from this picture is that many of these milk EV proteins, they cluster around certain hotspots, like the PI3 kinase, RAC1, CDC42. The other thing that we observed was that the blue proteins depicted here, the milk proteins, that they have a stimulating effect in this P38 dominated migration pathway, but that they have a strong inhibitory effect more in the proliferation pathway, in the MEC1-2 pathway. And that hinted us at the point that milk EVs probably influence this gap closure by stimulating migration. And in fact, when we performed this gap closure assay and analyzed the tracks of the individual cells, we indeed see that there was an enhanced displacement in the presence of EVs. And also, if we use synthetic inhibitors of the MEC1-2 pathway and the p38 pathway, as indicated here, you could see that only the p38 pathway could inhibit the effect of these milk EVs. So here, bringing together, bridging this protein cargo, we could explain that the milk EVs enhance the migratory capacity of the epithelial cells. The next step that we analyzed was whether milk EVs could also influence innate immunity. When a child is born, the gastrointestinal tract is exposed to many novel antigens, dietary antigens, microbes, environmental components. And there should be a way how this gastrointestinal tract and this epithelial layer can react in a controlled fashion. So we first used the HEC blue reporter cell lines to analyze whether milk EVs could influence toll-like receptor triggering. And what is depicted in this quite busy figure, but I will help you through it, is that if we incubate these toll-like receptor reporter cell lines, for toll-like receptor 2, with extracellular vesicles from milk, we could see that there was an activation. The same is true for when we look to toll-like receptor 4 reporter cell lines. Milk EVs could induce some extent of activation compared to the agonist. The milk EVs did not activate the TLR3 and 9 cell lines directly. However, you should pay attention to this point, is that the EV depleted control also showed some activation. So this could mean that it is not truly an EV mediated effect, but that it are components in milk that could be involved in the triggering of these toll-like receptor reporter cell lines, or in the procedure of the isolation of the milk EVs. What is even more remarkable is when we did the next experiment in which we incubated the reporter cell lines with their agonist in the absence or presence of EVs. You can see that for toll-like receptor 2 and 4, nothing happened, while for toll-like receptor 3, there is a very strong reduction of the agonist-induced response. That was also the case for toll-like receptor 9. So this is a very strong effect because we activated these reporters with the agonist, and EVs could override that effect. We then went back also to the gingival epithelial cell, the CA922 cell, and also if we activate these CA922 cells with poly-IC to activate toll-like receptor 3, this cell does not expect toll-like receptor 9, we could see that in the presence of EVs, the messenger RNA of IL-6 and CXCL8 was reduced. And looking to more genes, it was not only reduction of this IL-6 and CXCL8, but we also could see a reduction of several other genes like YUN and FOS. Remarkably, you could also see that there is a reduction of the TLR3 messenger RNA itself. So meaning that there is a regulation of, that there is transcriptional regulation. On the other hand, we see that some genes are really upregulated in the presence of these EVs, and amongst these genes are SIGR and SARM, which are negative regulators of toll-like receptor triggering. So that means that these milk EVs not passively inhibit this agonist-induced response, but that they also induce other genes that help to inhibit the response. And also here we try to connect the proteome with this observed phenotype. And that was not that easy because we saw these in blue, these milk EV proteins interacting at different hotspots, but that there were also hotspots down of the TLR2 and 4 pathways. We also saw directly activating proteins involved in the TLR2 and 4 triggering. So meaning that there could be a kind of equilibrium that is needed to get either activation or inhibition in the presence of EVs. Another remarkable candidate we found was the presence of cystatin B, which is an inhibitor of cathepsins. And these cathepsins are involved in cleavage of endosomal toll-like receptors to make them fully responsive. So when we analyzed whether this, so we confirmed that cystatin B was present in all milk EV samples, and if we analyze the full-length and cleaved toll-like receptors in this assay, we could see that there is indeed a reduction in cleaved and also in full-length toll-like receptor in the presence of milk EVs. So taking this all together, this means that these milk EVs, that they can regulate instantaneously by just interfering with these endosomal toll-like receptors, and also regulate gene transcription, which works, of course, a little bit later, which kicks in a little bit later. So then, as I mentioned in the beginning, these milk EVs, they do not only stay in the gastrointestinal tract, but they can also cross the epithelial border. And for that, we were also interested to see whether these milk EVs could inhibit CD4 T cell activation without, whether they could induce these, whether they could inhibit CD4 T cell activation. And that was indeed the case. We found that in the presence of milk EVs, the proliferation and cytokine production is strongly reduced. And here we could connect the milk EV proteome mainly to hotspots downstream of the co-stimulatory trigger CD28. So not on trigger 1 on CD63, on CD3. So since we found mainly these milk EVs proteins around this hotspot in the co-stimulatory pathway, we thought that maybe these milk EVs could induce Tregs, but we did a lot of experiments and that was not the case. In fact, we even found that when T cells were first activated in a primary stimulation in the presence of EVs and later on re-stimulated in an allogenic re-stimulation assay, they could be normally re-stimulated. So that means that milk EVs inhibit CD4 T cell activation without tolerance induction. And that brought us to this model. Importantly, what we try to tell with our work is that milk EV cargo acts in concert. So there is not one protein or there is not one microRNA, but they act in concert to target hotspots of signaling networks in target cells. And we show that these heterogeneous milk EVs, which carry these multiple components, can interfere in these three main processes: the epithelial barrier, the innate immune triggering, toll-like receptor triggering activation, and T-cell activation. So with that, we thought that maternal EVs delivered via milk create a window for regulated development of the GI tract and the immune system. And that brought us also to the next question is, and the question is, does maternal immune status affect milk EVs? So I just showed you that if we have milk EVs from healthy mothers, you can see that in the presence of these milk EVs, T-cell activation is inhibited, as we have seen it in inhibition of proliferation, inhibition of cytokine production. So what happens when the health status of the mother is altered? It has been shown that the sensitization status of the mother can influence milk composition, but that human milk and allergic diseases is still an unsolved puzzle. There is a very nice publication showing, for example, with ovalbumin, that administering ovalbumin via breast milk can inhibit the development of allergic diseases in mouse model. But if you use the same type of model and do not use OVA, but DERP1, another allergen from house dust mite, then the real opposite effect kicks in. So that is why there is still an unsolved puzzle. A couple of years ago, there was one paper that claimed that the phenotype of milk EVs varies with maternal sensitization, but until now, it is still unclear whether milk EV cargo and functionality are affected by maternal sensitization. So the question we wanted to answer is the T-cell modulation by milk EVs influenced by the allergic status of the mother? And for that, we performed the following experiment. We first selected non-allergic and allergic mothers. We isolated the EVs as described before, and we performed the T-cell activation assay on a primary CD4 T cells. As shown before, non-allergic EVs from non-allergic mothers, they really block the proliferation, and we do the same. We do see the same with EVs from allergic mothers. Although it looks like there is a little bit less inhibition of proliferation, but in general, they do the same. They inhibit proliferation. When we look to CD25 as activation marker, we see also that the EVs from allergic mothers go in the same direction and showing an inhibition, although we see that they are less activated compared to non-allergic mothers. Also for cytokine like IL-6 and TNF-alpha, we see we see an inhibition for the non-allergic. However, for allergic models, we see that IL-6 tends even to go up a bit in comparison to the EV-depleted control. And this is really a significant difference between the non-allergic and allergic mothers. The same holds true for TNF-alpha. And if we look to the NF-kappa B, to the nuclear factor NF-kappa B and AP1, we also see that there is a difference between non-allergic and allergic mother. Remarkably, also CDK4, we see a clear difference that there was a strong inhibition for non-allergic derived EVs, and for the allergic mother-derived EVs, there is even a kind of increase of CDK4. So in general, we see that the inhibition of T-cell activation is less pronounced in the presence of milk EVs from allergic mothers. And also here we try to connect the milk-EV cargo to altered T cell modulatory properties. And in this case, I show you the results and the analysis Alberto Giovinazzi did. And that is not on proteins. We look here to the other cargo, we look to small RNAs. And for that, you can see the workflow. Here we isolated these small RNAs and by analyzing these small RNAs, 33% of small RNAs appear to be microRNAs in milk. And we identified nearly 2,000 microRNAs in the milk. Then the next point, the question was, which are there microRNAs and which microRNAs are differentially expressed between milk EVs from non-allergic and allergic mothers? For that, we analyzed 714 microRNAs that fit this criterion. And in the end, we found 30 microRNAs that are differentially expressed between milk EVs from allergic and non-allergic mothers. Then the next step was to use a target prediction model of the differentially expressed microRNAs to predict strong targets. And then the next step is to perform Go analysis of T cell processes linked to these targets of differentially expressed microRNAs. And then now the process that is going on in the lab is to connect these processes to the observed T cell phenotypes. And I will show you some results of that. As I mentioned before, you can see if you look to IL-5, IL-13, and IL-6, and we can see a difference between non-allergic and allergic derived EVs clearly. If we look here to the signaling pathway that leads to the production of IL-6, IL-13, and IL-5, the STAT pathway is a dominant pathway. And indeed, we found that in non-allergic EVs, there are two microRNAs overexpressed that inhibit this pathway. So this could be the link between the cargo in the non-allergic EVs and the observed phenotype that we have in these functional studies. The same we try to do with the modulation of the cell cycle progression, because here we found that CDK4 has really an opposite effect in that non-allergic EVs, that they strongly inhibit CDK4, while the CDK4 is going up in the presence of the allergic EVs. And when we try to connect there, the microRNAs, it is clear that here are the ones are depicted of the non-allergic mothers, and in blue are the ones depicted of the allergic mothers, that microRNAs overrepresented in non-allergic EV target positive regulators of cell cycle, while the majority of the microRNAs overrepresented in allergic milk EVs target negative regulators of cell cycle progression. So what we see here is that these microRNAs, they present in allergic and non-allergic EVs can target cell cycle progression, but they can do it in a different way. So what did we learn now? By all these years of looking to breast milk vesicles and the different cargo and studying the language of extracellular vesicles. For me, one of the important take-home messages is that bioactive EV cargo hits hotspots in regulatory circuits at multiple levels. So it is not, as I mentioned before, one protein or one microRNA that tells the full story. It is the combination of all the cargo, of all the functional cargo together that clusters around important hotspots and ensures redundancy of regulation. So in general, I would like to say that the net effect of EV-mediated communication is determined by the crowd wisdom of deposited EV cargo at the target cell level. And that makes it very interesting, but also very complicated to study the language of EVs in breast milk. But now we start to understand a bit better these milk EVs. We are at the beginning, but we can also think on a biomedical application of milk EVs. And for example, I work at the veterinary faculty. We always talking about One Health research is by understanding these milk EVs, the role of EVs in milk, we can better optimize guidelines for early life nutrition. And that is not only in human, but also in animals, for example, for more sustainable livestock, coping with infections, reduction of antibiotics, etc. The knowledge that we also obtained from the studies of these different constituents of milk is also important to optimize baby formula and milk replacers for livestock, and also for milk processing of consumer milk. The next step is, of course, also the therapeutic application of milk EVs for immune disorders on regenerative medicine, or to use them as drug carriers, and also in infectious diseases like COVID. Therapeutic application of milk EVs may add to the full spectrum of possibilities to tackle complex infections. But before going there, I just kicked in here one slide of a paper that we published also this year to draw your attention, is that if you think on application of milk EVs, there is always the point on safety. The product should be safe. And that is also what, of course, for consumer milk was always the case. The product should, in the first place, be safe. So here I would like to show you some results to trigger your brains a bit about a comparison of raw bovine milk, pasteurized, homogenized milk, and ultra-heat treated milk, from which we isolated the different vesicles and first quantified the amount of vesicles or events by high-resolution flow cytometry. We could see here that when you compare raw milk with pasteurized and pasteurized homogenized milk, the amount of events does not really change. But we see a very strong reduction in the number of EVs in ultra-heat treated milk. So it looks like pasteurization does not have so much impact on the EV quantity. However, when we analyze with cryo-EM these EVs better, we could see that in raw milk, these EVs look very nice. While in pasteurized milk and pasteurized homogenized milk, we can see that the integrity of these EVs is really affected. In ultra-heat treated milk, it was very hard to really find extracellular vesicles that were intact. So also when we analyzed the RNA concentration of the same amount of EVs, we saw that there was a strong reduction in the amount of RNA already after pasteurization and homogenization when you compare it with raw milk. In ultra-heat treated milk, we nearly could detect, we could not detect RNA concentration. We could not detect RNA reliably, I should say that like that. In the Western blot analysis, it depends on the marker that you pick. For example, if you analyze CD9, we did not see so much effect on CD9 when we compared the different treated milk, the raw milk, pasteurized, and homogenized milk. Except the ultra-heat treated, we did not see any signal for CD9 and CD63. However, for CD63, you can see that there is quite an impact of the processing. So this indicates that industrial processing of milk can influence EV integrity and also maybe subsets of EVs, and that we should be aware of that. And that brings me then also to my overarching conclusion is that I think that milk deserves a more holistic approach, because raw or breast milk is not the same as the milk that we have now from the supermarket. And in general, processing habits are not primarily designed to maintain ingredient function. They are designed to make the product safe. And I think now to really have the full benefits of this remarkably rich and important fluid produced by nature, we need to develop new technology with maintenance of functionality without jeopardizing safety. And if we come to these new technologies in which we can keep the goodies in milk that mother put in there, we can really benefit the most from this rich and important fluid. And with that, I would like to finish and acknowledge all the people who were involved in these studies, and there are of course many people from my own group, but also the collaborations which are really instrumental to get all this work together. Thank you for your attention.
Thank you very much, Marca. It was a wonderful presentation. I learned so much about the milk EVs. There's a chat box there that if you want to ask questions, you can write it there, or also you can raise your hand and ask your questions. I have a few questions, but Clotilde's question, please do. Clotilde, please go ahead. You can go ahead.
Thanks, Marca. Very nice talk. Thanks, Carolina. I posted a question on the chat box. Actually, I got, and of course there was correction, automatic correction. That was not what I intended. So sorry about that. I wonder how the cystatins ended up being in or on EVs. Do you have any question? I suppose, well, theoretically, they should be on because they should be inside MVBs or inside lysosomes and maybe stick non-specifically to the EVs. What's your thought about that?
Yeah,
because
so
they are
that's
necessary.
Very good.
Yeah, sorry.
Yeah, so that's a very good point. So we did not investigate the topology where the cystatin B was, because that was indeed that is indeed an important point. If you really want to get these things together, you should prove that cystatin B can reach the lumen of the endosome to interfere in this processing of the toll-like receptor, and that we did not perform. We did not look to the topology yet, and it is something I hope we can go more into detail to really explain or to see whether this cystatin B could play a role in this altered cleavage of toll-like receptors. But we observed indeed less cleaved toll-like receptors, TLR3, in the condition in which we have incubated the assay, in which we added the EVs to this assay. But you're right, we did not fully prove it.
And could this mean that the EVs that you recover from milk are of late endosome, lysosome origin, or maybe not necessarily because, I mean, I don't know.
yeah, I think that is also something which is really important, is to see how we can now trace back what is the origin of these EVs. So not only from the cells, but also whether we can trace them back to the, whether they maybe are derived from late endosomes, or, but for that, we really need to go into the detail and see how they are formed. But the first question is, that we need to figure out is, where is the cystatin B containing EV coming from which cells? So that is the first step we are doing now, is to see what are the main contributors to the EV pools. And the interesting point is that most people think always that are the epithelial cells themselves. But in fact, when we did now the first analysis, and that is Martijn van Herwijnen is doing that, he is trying now with the bioinformatics approach to see whether we can find specific signatures of cells back on these EVs to see which is the cell of origin. And what we all the time get back is that most abundant EVs that we can trace back come from immune cells and not from the epithelial cells. So there are, of course, epithelial-derived EVs in there for sure, but I was really surprised by the high number of hits that we have from T-cell-derived, neutrophil-derived EVs. So that is really the first thing to do. And if we then find the cell of origin, I think there we can also look to where are the EVs coming from, and are they released while the cells are in milk? That's the other question, because neutrophils, T cells, they are all living and they are in milk. So are the EVs released while they are in milk, or are the EVs coming from cells somewhere else in the body and are transferred into the milk? That's another puzzling question.
Yeah. Thanks a lot. Thank you, Marca.
Very interesting. Genevieve, please go ahead with your question. You're still muted.
Sorry, I was trying to unmute myself. Hello, I was curious to know how you did show transfer of milk EV through the gut epithelium, or did you not?
Yeah, so I personally did not show that. So in my group, so I refer to literature in which it has been claimed that these EVs can transfer via the epithelial cells, and that you can trace them back in lymphoid tissue also underneath, or that you can trace back, for example, if you look to in bovine milk, there is quite some work done with specific bovine microRNAs and bovine proteins that you can find back then in different organs after oral ingestion of these EVs. So I did not show the results, and I did not study it myself, but that's what has been claimed in literature.
Okay. I actually have another question, which is a bit more about your the cow's milk. I know a lot of people, including myself, cannot drink this natural milk. That makes me sick. So could that be explained by the EVs in the milk?
That's
because now you show
point.
that the natural milk would be better.
Was that the whey proteins or maybe lactose?
Yeah, so the natural milk, I'm a bit hesitant to say that the natural milk is always better, because there is a reason why we pasteurize milk, and that is that raw milk, if you have raw bovine milk, there is a safety issue, of course, and that is what I should say first. So I do not want to say that everyone should now directly go to the raw milk. And why you cannot stand raw milk can have many explanations. And I don't know whether that are the EVs. I think that would be very focused and narrow-minded to only think on EVs. I think there are so many components and so many general rules that it is problematic to drink raw milk. And the same holds, for example, true in the discussion also with human milk is, of course, when the mother nurses her own child, we give the raw milk, and that is also accepted, and that is what we should do because that is how it is designed, the whole system in nature. So that's breast is best, mother's breast is best. But then the question pops in, if the own mother cannot feed the child and you need a mother milk bank, what are you going to do? Are you going to make a very safe bank? Taking the precautions, for example, we also think about cow's milk and things like that. And I think that is now a big discussion. Do you need, so there is also a risk to give the raw milk from a donor mother to another child. So I think there is really the gap I would like to dive in from scientific point of view is like, how can we think on a safe product in which we can preserve the goodies from the mother? And that's a long way to go, I think. But the discussion is now out.
Thank you.
We have questions from Antonio Marcilla, which is quite interesting here, and he asked me to ask to you. So have you analyzed milk EVs from mothers that are allergic to cow's milk?
I have to think whether in this group there were maybe some cow's milk allergic mothers. They should be in there, but we did not specifically included that group because we wanted, I think we excluded them in this study because we wanted to have a non-seasonal, non-dietary allergy to have a more constant group. So this is more the house dust mite we all face all the time and things like that. So the answer is no, we did not specifically look at that group.
Yeah. So I myself, with having a toddler that's still drinking formula milk, so I was just wondering, because you haven't really showed the dry milk powder, can we actually reconstitute EVs out of dry milk powder? Have you looked into that sort of thing?
Yeah, we have looked into that a little bit, also with Friesland Campina, and you can reconstitute particles, something like that. But I am very reluctant to say that you are reconstituting a fully functional milk EVs, and as I showed you also already with the pasteurized EVs, you could see that there is an impact on the integrity and an impact on the RNA cargo. Yeah, that is really a complicated story. But I know, so you can reconstitute particles, but yeah, how far they really look like the raw milk EVs, I'm not sure.
And how about metabolites or small molecules like probably vitamins and that sort of thing? Are they being carried by EVs?
Excellent question, Carolina. That's where we are now really starting in, and we have now done the first measurements. So we are really now in the metabolomics, and the lipids. So we have done the proteins and the small RNAs, but the metabolites are very interested. We are very interested in that because what we have observed, these effects also in the migration assay, they are extremely rapid and they are transient. So we are very puzzled that if if you think that the regulation is more around gene transcription, etc., that is not that fast. So it can also kick in, but there is something else going on that is ready to act. And so that is why we look to the proteins first. And that is why we look to the metabolites now, because that are all the things that are ready to act. And we see these instantaneous effects. So I hope to give you an update on that within half a year.
Yeah, that would be really cool. And I think my last question is actually about the size of this milk EV, which is like you're saying also like it is about 200 nanometers. So it's really much, well, almost double the size, I guess, with the average size we found in plasma. So, but then in terms of markers, you show that this has CD9, CD63. So I don't know how to get them together now in terms of the biogenesis and, you know, the secretion of these vesicles into the milk. Have you actually looked into sort of intracellular derived marker like the TSG101 and ALEX? Do they also have that, or is it just mainly like transmembrane protein? What do you think?
No, I think we also had TSG101 in there, but I think so. So the main discussion is about the size, and you can see that there is indeed they are very heterogeneous, and the average size, as measured with NTA, looks around 200 nanometer, which is bigger than we measure also with the plasma. But the question is, what is the actual size and how do you measure it? I think it also has to do with the composition of these EVs. And what we have seen is that milk EVs, they are also very rich in mucins, and that are very big molecules on the surface. So yeah, so it depends on what is the core diameter and what is then around that corona of these EVs. So I think there is the main difference in these milk EVs, that they are extremely heterogeneous, and that we see also these very huge proteins that can be exposed on the surface of these milk EVs, that maybe also affects the size, but also the method that you use to determine the size makes it also different. Like this is then measured with NTA, but if you just measure just the from lipid bilayer to lipid bilayer, you can come up with some other size ranges. So the orthogonal methods also for this to really determine size is already very important to say something about whether the size is so different or not.
Yeah, there's still a lot of things that we need to do in terms of how to get the really dependable methods to measure the size of vesicles, I think. So exactly. Yeah, thanks so much, Marca. That was a very wonderful talk, and I really enjoy also the discussions. Thanks everyone who's asking the questions as well.